Welcome everyone to the African Doctoral Academy Masterclass in collaboration with the African Microbiome Institute. Today's masterclass is going to be focused on an introduction to the microbiome and the importance of multidisciplinary microbiome research in Africa. I am joined by Dr. Kristen Neal van Zyl. Dr. Christian Neal van Zyl is a computational biologist at the African Microbiome Institute, Division of Molecular Biology and Human Genetics, Faculty of Medicine and Health Sciences at Stellenbosch University. Dr. Neal van Zyl completed her undergraduate studies in molecular biology and biotechnology at Stellenbosch University before advancing to the Division of Medical Microbiology at the institution for her postgraduate studies. There, she actively participated in numerous multidisciplinary research projects and earned her BSc and MSc degrees, both with distinction. Since 2017, Dr. Neal van Zyl has specialized in microbiomics, focusing her PhD and postdoctoral research on the impact of levofloxacin on the gut microbiome of children in Cape Town communities. Dr. Neal van Zyl specializes in microbiogenomics and microbiomics with a focused interest in teaching, training students, and building bioinformatic capacity in local and African research spaces. In her current role as a computational biologist with the AMI, she provides microbiome analysis consultations, trainings, and workshops, aligning with her commitment to education and research development. I am now going to hand over to Dr. Christian Neal van Zyl. Over to you, Doc.
Thanks so much, Luther, for that kind and thorough introduction. I'm very happy to be here today. For those who are looking to contact me, my details and those of the African Microbiome Institute, who I'm currently employed with for the last couple of months, which have been great. That will be at the end of the presentation. So for the rest of the presentation, I will be turning my camera off just to get some extra bandwidth. Yep, let's get going. So hoping you can hear me clearly, I'm going to move a bit closer to my mic. Okay. So let's just get into it. So I'll give you a quick overview of all the topics that we are going to cover today. And I'll try and get through them relatively quickly, just so we have more time for the Q&A at the end. So first, we'll go through some basic definitions, take you through how microbiome research is multidisciplinary, some concepts that you need to understand, key parts of study design, and research challenges we have in the African context. So moving on, what is a microbiome? Well, basically, a microbiome is where you have an environment, and this could be a human site, so your skin or your gut or your lungs, soil, plants, roots, marine environments, or even animals. You need the microbes, so all the bugs, so to speak, that live in that environment. And you also need their DNA and any metabolic products that they might be producing. And all of those things together and the effects on the environment, that system, that entire system, we consider to be the microbiome. So that's a bit of a more condensed way of putting the microbiome definition. And I also want to focus your attention to the top definition there, which are the microbiota. And those are kind of the bugs that I just mentioned, the microorganisms that are present in the environment. When we were just looking at them alone, we would call them the microbiota. A lot of people use these two terms interchangeably, but actually that's not correct. We also have the metagenome. So that's when you take the DNA of all of these microbes and you put that together and you take all of that together. That is called the metagenome. And that may also include, let's say it's a human microbiome, that would also include the human DNA and not just the microbes. Moving on, all of these have meta in the name, as you might think. The metabolome is when you look at all the metabolites that we have in a system or a single tissue. So some people have two definitions for them, but the most common one you'll come across is the metabolome. We also have the transcriptome. So that's when you no longer are looking just at DNA, but you also look at RNA. And these are kind of things we can look for function. You'll see that in the next slide. And we also have the proteome or the metaproteome when you consider a bit larger molecules than just metabolites. Sometimes these are interchangeable, but if you're looking at something very specific, people like to have their definitions. So it's sometimes just good to have them in your back pocket. So why is microbiome research considered multidisciplinary? Well, that's because we can do a lot of things with microbiome research, and we need to look at it from a lot of angles. The first angle a lot of people start with, and the one that we'll also be focusing on in this masterclass, is the microbe angle. So looking at the microbiota and their genomes, and we can do association studies here. We can look at, for example, Luther mentioned in my bio as looking at the effect of an antibiotic on the microbiome. That's an example of an association study. You could look at antimicrobial resistance. We can also then look at the functions, and that's where these other parts of the definition start coming in, where you look at your metabolites, your expressed RNAs, so your transcriptome, and your proteins. These give you more information about function. You can also sort of link the metagenome to function by predicting it, but it's not as good as having the actual metabolites themselves. Also, in terms of function, you can start asking questions about immunology, especially in human and animal systems. How does the microbiome connect and interact with the immune system? Because there are interactions. We can think about drug discovery if we're looking at metabolites. And now you can already see we're going in multiple different directions. It might already be a bit difficult to keep up. And then we want to think about all of these things in a One Health perspective. So how does our environment and our agriculture feed in with our humans? How does the humans feed in with health? And how does this little circle go on? That is kind of a One Health perspective. So let's say we want to take this into policy. So we want to make actual changes. We want to do translational research. We don't just want to see what's there. We want to see how can we use it to better our society or our environment. Then actually we need a lot of different perspectives. And this is where the multidisciplinary nature comes in. So let's take, for example, we want to take a microbiome diagnostic. So we want to use microbiome in determining certain disease states. And now we want to put that into the system. We want to actually offer it to people. Now you need to think about all the stakeholders that need to get involved to be able to put this into place. One you might think of is the Department of Health, but they're not just a biomedical sciences stakeholder. They're also a political stakeholder. They could be an economic stakeholder because we need to know how much things cost. They could be engineering stakeholder if we need to figure out how to make a specific machine or make a specific chip or tool. So you can see here that we have a lot of people interacting to actually take our microbiome research, make something tangible happen. You don't necessarily need all of these sciences or stakeholders involved, but it does take more than one. And that's where my next point comes in. One group cannot do it all. And this is where microbiome research is very, very much rooted in collaboration and starting small, building up steadily, and then also thinking about how you can use your collaboration to build capacity elsewhere. Because one group can't do it all, but we might be able to learn from each other and implement things. So, for example, collaboration is not just people from different departments or different faculties working together, or maybe people in the same research field, but the one has a better system or has more knowledge, and they teach the other to then do it in their own setting. So this, as my first point rightly mentioned, this multifaceted nature makes microbiome research very beautiful, but it also makes it very difficult. So to not get lost in this system, we really need to formulate our research questions very carefully, because this will take us all the way from sample collection to data analysis. And we can never just, in microbiome research, for example, if we want to sequence, it's never just because in our context, money is often a very big limiting factor. So we don't just do things because they exist. We need to know why we are doing things. So, like you may have seen from my first couple definition slides, the microbiota is often the starting point of many microbiome studies. It's one of the things that you might think of immediately when you hear the word microbiome, and most of the research around the microbiome is based on microbiota. Now, this could be done through culture, where we actually take organisms from an environment and we grow them, or we can look at them through microscopes. We can do targeted sequencing where we want to look at specific microbes, or we can move into the metagenomic range where we try and sequence everything in a sample to study them all together. Because sometimes taking things out of the system, you can study them individually, but that does not mean they interact the same way when they're in the system. So for today's session, to keep from losing ourselves in the microbiome system, we're just going to tackle some basic microbiota analysis concepts. So here's where the study design comes in. First, we have to ask ourselves the question, which microbes do we want to study? Because we get lots of different kinds. So in most human studies, bacteria are your main topic of interest, and that's mainly because they dominate the human systems. They're present in the highest abundance. In many soil studies or other plant studies, you might see fungi come up more often because in those environments, they are more important or perhaps more abundant, not necessarily important. So you want to know, what am I looking for? And then why am I looking for it? Am I trying to describe what's there because no one knows? So that's a valid question. Or we sort of know, but we want to know how it's affected by something. So that could be intervention. We want to give a person an antibiotic and see how their microbiome reacts. Or we can do association studies where we try and figure out why the microbiota are the way that they are in different systems to see what makes them the way they are. So to do a microbiome study, we of course also need to consider what our sample types is, how much we need, how we can collect it, and how we need to store it, because all of these factors can influence the quality of your nucleotides or your DNA, which is what you'll be needing to sequence afterward. So we need to know kind of what do we expect in our samples? Do they have high abundance of the microbes we're looking for? Are they easy to extract samples? Or are they low abundant samples that might be more difficult to extract? I'm going to again use the human system here as an example. If you take a specimen from a human, often there's a lot of human DNA in that specimen, and it can overshadow the microbial DNA. So that might be something that you need to consider later on in your workflow. You also need to collect lots of data, because without data, we won't be able to do any of the association studies that we want to do. And we also need to consider contamination, which is a major factor in microbiome studies that we need to control throughout. After we've got our samples, we figured out how quickly we can get them to the laboratory. Can we freeze them? Do we have capacity for that? We need to actually do the nucleic acid extraction. Then we need to decide what kind of sequencing we're going to do. And often this is very much limited by cost. So what can we afford to do? But otherwise, it's also dependent on what kind of questions you are answering. So if you just want to know about the microbes and their associations, you could do a normal targeted type of sequencing, targeted amplicon sequencing approach, which is what you commonly see out there in the literature, what people do. If you want to know a bit more about how they function, you need to do other kind of tests. And that's where you need to consider your sample collection and storage. Because, for example, one thing that we might want to do is if we are collecting samples in a field, we're taking them to a laboratory, and we want to know that our DNA is surviving, we might want to put a DNA stabilizer in that sample. But often DNA stabilizers will kill anything off that's still alive in the sample. And then you can't do any kind of culture work. You can't grow these bugs again. So then you're limited by what kind of questions you can answer. The stabilizers might also affect metabolites in your sample. So if you're wanting to do metabolic studies, you might not want to do that to your sample first. So this is why you need to consider your entire process when you are actually at the start. What kind of question are you asking? The last point that you'll see here, or the last little arrow, is data analysis. And that obviously very much is informed by our research question and the kind of sequencing data we have. So we can only analyze what we have. We cannot pull something out which does not exist. This might seem like a very obvious statement, but it's sometimes something people forget when they're starting off planning for such a big study. Okay, so the stars, they denote places where we often still have difficulties in many laboratories throughout our continent. So this is to do with sample collection logistics, especially if we are collecting in communities or environments that are far away from the laboratory, and our transport systems are not necessarily ideal. So that logistics might be an issue. Data storage might be also a big issue. The actual sequencing might be difficult to do depending on what is available in the setting. And the data analysis takes lots of computing power, and that could also be one of the little bottlenecks for the African population. So we'll get more into these challenges a bit later on in the presentation. So like I mentioned, today we're just going to be tackling some of the microbiota sequencing techniques that are out there. So in my eyes, the three big ones would be whole genome sequencing. And whole genome sequencing can only be done on isolates, really. So the thing here is you cannot have really a mixed sample and do proper whole genome sequencing. So this isn't really a microbiota technique per se, but if you have lots of resources and you are targeting specific bugs in part of another study, you might consider this as part of your arsenal. The second one is for sure the most common sequencing technique used to analyze microbiota, and that's targeted marker gene sequencing. So here you might have heard this before, 16S rRNA gene for bacteria or archaea, and the ITS region for fungi. So these are probably the ones that you would, or I would think of when I hear the word microbiome study. And the third point here, metagenomic sequencing, sometimes we refer to this as metagenomic next generation sequencing, or MNGS, is something that's becoming more and more common. It is definitely more expensive to do, but you do get a lot of more information out of your sequencing than you do with targeted. So these two that I've put the boxes around, these are kind of our well-established sequencing techniques currently in the field. So I've put a little environment here. So let's say we are looking at the hand and we're looking at the skin microbiome. Some questions that we might want to ask ourselves is first, which microbes? So which ones are on the skin? Which ones do I want to look at in the first place? Or something like an association, like I mentioned earlier. Which ones stick around after washing? So the which microbes question, the concept that we're tackling there is called abundance. The next question we might ask is, okay, we know what's on there now. How many? How many different kinds? How many are there of each microbe? How many are there before and after washing? And this concept we call alpha diversity. So the more official definition for that is the richness and diversity of microbes in a specific system. But that is basically how much and how many. The third question that we often start off with in our microbiome studies, how does this community, so the microbial community, how does the community on this hand skin compared to my other hand skin or some other person's skin? Are they the same or are they different? How does it compare to skin that's been washed with soap? So the actual dissimilarity of microbial communities we refer to as beta diversity. So those are kind of the top three things when I see a microbiota study and I start reading. These are the three things that I usually expect to definitely be there, as at least as a starting point. Some things that I would consider if I'm doing follow-up after my initial investigations is firstly, how do the microbes interact with each other on the hand? And you might think of something like correlation. So to see which bacteria are present together or which ones are usually not present together, or we can do network analysis. We might want to look at the function of the microbes because sometimes just knowing what's there doesn't really give us enough information to take the study forward. We want to know, okay, what are they doing? And if we introduce a change in the system, do they do something differently? Or can they do something differently? So we can do this with sequencing through something called predictive metagenomics, at least DNA sequencing, which is the one we're focusing on in this topic today. A third question you might want to ask yourself, are these microbes resistant to medicines that I'm giving or chemicals that I'm applying? And that we can do through resistome analysis. Okay, but now this is where the study design matters. So if we had done targeted 16S rRNA gene sequencing to detect bacteria, which of these three follow-up areas would we have been able to explore? And that would be the first two, because we can actually use our bacterial information and we can plug that into very nice algorithms that kind of predict what their metagenome would look like, what functions they can do. So this isn't necessarily as good as the real thing, but we are moving towards that prediction. What we can't do is resistome analysis. Because we did targeted sequencing, we would only be targeting this gene, and this gene would only be able to tell us which different kinds of bacteria are in there. Because this gene is in all bacteria, and it has conserved regions where you can put primers to amplify it, and the bits in between those conserved regions are hypervariable, which means you can use the hypervariable bits to distinguish between different genera or species. But because we didn't do any other sequencing of the genome, we don't know anything about their resistance genes because this doesn't apply to function. Now let's say we didn't do targeted sequencing, we did metagenomic sequencing. Which of these areas would we be able to explore? Now we should be able to do all three. Of course, this does depend on how good your sequencing is and how much you were able to pick up. Sometimes we see, especially in human studies, that we can see what's there, but we struggle to see what kind of resistance genes or mutations they have, especially in samples where you have host DNA that's taking over the entire sample. It can be difficult to generate enough microbial DNA. But in principle, this is why your design matters. It matters which kind of questions you want to ask, because that's the techniques you need to then choose to do if you have the funding to do them. Another thing about metagenomics, and you would see that pop up here, it's more than just bacteria, which a lot of microbiome studies focus on. So with metagenomics, because you're sequencing everything in a sample, you should theoretically be able to pick up other microbes like fungi or viruses, protists, anything that you have extracted successfully the DNA of, you should theoretically be able to pick up. This does, however, become difficult sometimes as we don't have very good reference databases existing for all of these microbes. But as we generate more information, the reference databases get better. So now we're coming to some of our last points before we move on to the questions for today. That is the research challenges that we face with microbiome studies in the African context. So again, because this is my main field of expertise, I'm using the human microbiome study as an example. There was a very lovely systematic review published in 2021 from colleagues from UCT, where they looked at human microbiome research throughout Africa. They found that only 60% of African countries were represented with samples. 80% of the first or last authors, which are in, at least in microbiome research fields, mainly the two primary positions for authors to be in, they were not from African institutions. And also that the funding for microbiome studies comes from outside of Africa, which is not surprising. US and EU is leading there. They also found or published or showed some research from other studies done in the Southern African communities that found that only 3% of intra-regional and 5% of continental collaboration was present in countries in our area, in contrast to 47% of studies having intercontinental collaboration. So that was showing that within our regions, there's not a lot of collaboration, at least at a couple years ago. So this means that many research projects are siloed. They are happening in isolation, which means they don't get as much reach as some of the studies that have collaboration would get. So why is this representation issue a problem for us? And that is because Africa is the second largest continent by both size and population. So that means in terms of the global microbiome research that is very much focused on the Northern Hemisphere, we are severely underrepresented. Because we have so much geographical and ethnic diversity, this is kind of a mine of information that we haven't yet delved into efficiently and that we need to know if we want to move into translating what we know about the microbiome into practice. The reason I bring this up is, for example, a very hot topic in the field is personalized medicine and using microbiome sequencing of individuals to be able to tell them information about their health and how to change their microbiome to benefit their health. We can't do this in a lot of African countries because we don't have a lot of microbiomes. So we don't actually know what our populations look like. And microbiomes differ vastly between different populations. So before we can even move into that kind of scenes, we need to know what's there. We also need to consider things like climate change, which will greatly affect our food production and things across the continent. And this is where the agricultural sector and environmental microbiome research is very, very important because we need to know what's there and how it will be affected if we are to know how to best remedy the situation in the coming decades. So we've gone through a bit of the underrepresentation and silos. Now I want to bring you back to the 3 stars that I showed you on the study design, where I mentioned that logistics could be a problem, sequencing could be a problem, and data storage and analysis are still factors for us. And this is because of sample collection, where you take place, where this takes place and how far from the laboratory can be a big issue. Because often to keep our sample integrity, we need to be able to store them in cold chain, or we want to do more than just DNA sequencing analysis. In other words, we have samples that we can't stabilize and transport at room temperature. That or it would just be extremely expensive sometimes to transport samples to other places to do, for example, DNA extraction. And also we have to consider things like material transfers. If we are transferring samples to other countries or other institutions, there's a lot of red tape in the background that's difficult to set up sometimes and also expensive to send. For the sequencing itself, the type of sequencing that you can do is very cost dependent, and this is why it's a limiting factor for studies. It's still relatively expensive compared to many other methods, even though the cost has come down significantly in the last decades. Per sample, sequencing can often be very cheap if you have lots of samples, but that means you need a good chunk of funding to be able to do lots of samples. And if you don't have lots of samples, it can be very expensive to do them. And lastly, like I mentioned, I'll just retouch on it. Data that you generate from sequencing is very large, and we need to store this securely. And doing that in a lot of places where we don't have good resources or infrastructure set up for that can be difficult. So what are the solutions for these? So what are we doing? By no means is this a comprehensive list of efforts and strategies. I'm just going to bring up a couple examples to show you that we are not sitting back and letting these challenges persist. So in terms of siloed research, well, us at the African Microbiome Institute, we are actively trying to combat this by working cross faculties, trying to get people together, having a network, having people talk to each other, share their expertise. There are also other groups, and I'm going to highlight the H3 Africa Microbiome Working Group because they have just published a lovely paper where they are trying to make a database for South African microbiomes and African microbiomes, which is one of the problems we mentioned before in terms of underrepresentation. And there are more. So there are definitely efforts to try and get people together, make sure we have all the expertise we need to do these multidisciplinary studies, and also to do training. We want to increase the capacity of labs across the continent. For logistics, I've just put two things here. The one is the stabilizers. I've mentioned them a couple times. This really has improved over the last couple of years. It really allows us to keep samples for up to a month or more, sometimes at room temperature. This can mean a lot for number one, bulking samples and keeping them stable, knowing that you won't destroy the integrity of your DNA before you get to sequencing it. So the improvements in these kind of stabilizers over the last couple of years have been amazing for logistics. Another thing I'll bring up is the rise in popularity of the portable sequencers. So I've put the Oxford Nanopore Mac1B sequencer here. This sequencer is actually palm-sized. So it will fit in the palm of your hand. It can get plugged into a laptop. So if you have a small power supply, you can actually take the sequencer out into the field and do your sequencing right there and then. This is also great for smaller labs because it's reasonable in price. So this is a step up for many people who haven't previously had access to bigger sequencers like the Illumina sequencers, which are amazing. But it's just another bit of, advancement in the field that's really taking us up a step up in logistics. Moving on to sequencing capabilities in general, across the last, I would say, 5 to 10 years, we have gotten a lot of access to sequencers in many places, both locally, so in the Western Cape, and nationally in South Africa, and also, of course, in other countries, but I'll highlight the South African ones for now, and just the ones here in our hood, if I can put it like that. So we've got Illumina sequencers at CERI, we've got Illumina sequencers at CPGR, at UCT, at other groups in the university, and there's an ion torrent at the Central Atlantic Analytical Facility at Stellenbosch campus. There are big sequencers, both Illumina and others at the NICD, and also Inkaba Biotech has a PacBio machine. So where we used to not have access to these machines, we have more readily available machines around us now, and a lot of the labs. So these are mostly service providers that I've listed here, but there are also labs that have these sequences that might be willing to collaborate. And this is where we can now start using this network that we have to put people together to actually do the sequencing and resolve our challenges. Lastly, just a touch on the data analysis side. So we've recently gotten access in Cape Town. At least Amazon has moved in their warehouse cloud solution system, so people can actually pay to access their cloud computing software, although it is extremely expensive. We also have more and more access to high performance computing servers throughout South Africa. The CHPC is one, but Stellenbosch University also have them, multiple of them. So we are moving up in terms of having access to these things. We just need to look around a bit, browse our network, and see how we can get things done, not just necessarily on our own, but together collaboratively to move forwards, and push microbiome research in Africa towards translation. So that's my speech. If you have any questions, please do use the Q&A function. We'll be answering them here. The QR code that I've linked on screen is actually to another ADA AMI collaboration where we'll be doing a 5-day hands-on targeted microbiota analysis workshop at the end of June. So if you're interested in that, please scan the QR code and it will take you to their application form. Thanks.
Okay, thank you very much, Dr. Christian. We do have a few questions in the Q&A, and I'm just going to read them out and maybe, yeah, you can address them. So the first question that we have is from Stephen. And Stephen is asking, what do you think about the possibility of establishing poop banks to explore the diversity of African gut microbiome and to use in poop transplants to reset the gut?
So that's a really good question, actually. I like the idea of them, because it is something that has been done internationally. So I recently attended a European conference, and one of the talks there was actually similar to this, where they had created kind of they tried to see if there was a golden bullet, so to say, in terms of your fecal microbiota, to see if there are better ones to be able to transplant, to reset people who have been sick specifically. So not necessarily a non-sick person, but someone who has been sick, to try and see if they can restore their gut microbiome. Happens often in people who have Clostridioides difficile infections. So if they get severe, sometimes you're hospitalized, you get a lot of antibiotics. It creates a niche for this organism. It takes over and you get extremely sick, and often antibiotic treatment does not work. And then they do these fecal microbiota or poop transplants. The problem with what they've seen internationally is the reversion rate. So it seems to work for a while and then it reverts back. So the rate of which it actually sticks around to do that reset is not yet clear. So internationally, it's not clear yet. So that might mean that it might be difficult for us to do here as well. But it certainly is something that we need to think about. It should be similar to how H3 Africa has been trying to do the human biobank. They've been trying to bank samples similar to this, so that we have a resource. So, I mean, I'm definitely in favor of that. I think we are moving towards that direction. We're not quite there in terms of the transplants, but, all in good time.
Okay, we have another question over here. How much does it usually cost to get access to the CHPC? I particularly need it for molecular dynamics simulation.
So I believe if someone else knows a bit more, they are welcome to jump in the chat and also let us know. I believe that Stellenbosch University is a partner of the CHPC, and I think some other universities as well, and that you can actually, as a group, apply to use the CHPC. Um, I don't think there's necessarily a cost, but I think there's an application process. I could be wrong. For the Stellenbosch University HPC, there are registration costs involved per year, um, but it's very affordable, very, very much affordable. So yeah,
Okay.
if someone else knows a bit more about the CHPC process, please do correct me.
Okay. We have another question. Please explain more about the relationship between the gut microbiome and brain health relationship.
Yeah, that's another very interesting one again. Um, I'm not going to go too deep into it because I'll actually point you to some of our experts at the Faculty of Medicine and Health Sciences, the Neuropsychiatry and Genetics group, led by Sean Hemmings. They actually do a lot of this kind of research. So this is very much their field, but I'll just dip into it briefly for those who don't know. So there's something called the gut-brain axis. And there have actually been a lot of studies that have found associations between changes in your gut microbiome and whether you are more likely to have certain neurological diseases or syndromes. So, for example, developing Alzheimer's later in life. Or with autism spectrum disorder, there have been a lot of association studies. So a very important thing that we need to note is many of these studies have just found the associations. They have not necessarily made causal links yet. So they know it's related, but they don't know why or how always. But like I mentioned, this is not my field of expertise, so I will type it in chat, the group that does this kind of work. So I will put their information for you if you want to go read up some of their work.
Okay. We have another question. What are the potential unintended consequences of manipulating the microbiome through interventions such as probiotics and antibiotics?
That's a very, very, very good question. So yeah, it comes back to the fact of having information. Um, so we don't have a lot of studies who have actually done this translational work, long-term work, to see what are the long-term consequences of some of the things that we actually do to the microbiome. If we now do a fecal microbiota transplant or we give a probiotic, the research is still very, very new. In terms of the scope of all research out there, microbiome research is still very much new, in my opinion, at least. So
Okay.
we haven't had the time yet to kind of do these kind of studies, but it is certainly a risk.
Okay. Another question. How do we balance the need for scientific progress in microbiome research with the ethical considerations of studying human subjects?
Yeah, that is a lovely question. I've recently had um, talks with someone. He actually did. It was a professor from, well, he's mainly at Pathcare, but he's also at UCT, and he actually did a big talk on microbiome ethics recently, and he spoke to me about it before. So it's really about thinking about the overall benefits here. So obviously we want to be kind and ethical to our participants, and we kind of need to think about how this would benefit humans and also the participants in our studies. So not just doing a study for the sake of getting the information, but also thinking about how can our research actually benefit the people that we are getting samples from. And that's where I feel like the balance in terms of ethics comes in, is you want to ask the kind of questions that can give you useful information that you can then feed back to that community. And it might not affect them directly, but it might be useful down the line. So I think where we need to balance it is to make sure that we're not just collecting from random people just for the sake of it, unless we are doing a major call for volunteers. But we need to make sure that our communication, our scientific communication, is very much on par, and that we always keep the participants in the back of our minds as to how can we help them, how can we educate the people in general. So I think that's where we need to go in terms of advancing microbiome studies, is bring the people with us, bring the participants with us along the study, make sure they get feedback, make sure they understand what's happening and how it's benefiting them. And of course, that should be, it's something I've actually added to the workshop that I'm doing in June. A couple slides that I've added is the ethical considerations that we need to keep in mind. Those need to be there with your research question from the start.
Okay. And we have a comment, rather. I'm sure the participant would like your opinions on that. They say, I have learned from one of the microbiome specialists that sample processing and analysis should always be performed with negative and positive controls from the DNA extraction to the sequencing and microbiome analysis. So perhaps your comment on that?
Yes, I absolutely agree. And I think he might have learned that from me because I know who made the comment. So it's something that I recently enforced, um, is and something that I have not necessarily done well in the past. When I've published my research, I've always had these controls going. So this comes back to the point that I made in the presentation. If you will recall, I mentioned that contamination is a big issue. So this is where these negative and positive controls come in. We want to make sure that what we are seeing when we are sequencing a sample is what's actually in the sample and not something that we actually accidentally introduced. And that's because, especially when we do something like bacterial sequencing, it amplifies all bacteria. So if you accidentally contaminate your sample, it will also amplify the contaminants. And that's why you have negative controls that you take with you from the start, because then if you do see contamination, you can either try and remove those sequences in your analysis. There are ways to do this. Or you might see actually your contamination is quite low, which is usually a good sign. He's also mentioned positive controls, and those are really important. Again, because we want to know that what we are seeing is legitimate, and that's something usually where we'll put in a mock control where we know what is in the mock. We'll choose a couple of bacteria that we know how much we've put in and what we've put in. And then once we've done our analysis, if we analyze that mock sample and we see those bacteria that we've put in, we kind of know our pipeline works. So yeah, I definitely agree. Controls are extremely important in all microbiome studies, not just sequencing.
Okay, we just got another question. In terms of overcoming the silos in which this work is done, how would someone who is in the social sciences get involved? This person goes on to say, I am part of a project that is working on the use of population descriptors in microbiome research, among other issues, from an interdisciplinary perspective that I am sure would also be interesting in the work being done here at Stellenbosch University. Through what channels would collaborations be possible, or how could I help connect researchers with each other?
Yeah, so I mean that's basically what the African Microbiome Institute is. One of our core functions is that we try and connect people to get together to do these interdisciplinary studies. So because those are the kind of things that kind of help us get funding, competitive funding, where we have bringing a lot of different experts together. So you can certainly contact myself. Um, I see now I didn't actually put my email on this last slide. Apologies, I thought I had, but we can just post it in the chat quickly, so that you can get into contact with me, and then we can put you you can get on the African Microbiome Network, which is how we distribute all our events and things and how we get together. So what we also do is we have regular scientific meetings where we actually encourage studies like yours to be presented. So that's something that you might be interested in. So when you connect with me, I will let you know when our next meeting is. Maybe we can, get you up to present there and have you then directly put in touch with members from our network. So there are also other considerations. I mean, you're saying that you're from um, political sciences, and that's exactly the kind of people that we also want to get involved, because obviously as um, biologists or scientists, often, we don't get necessarily those who are not working with human subjects directly don't get that perspective. So it's important for us to bring people from other faculties in to help with that. So please do get in contact with us. It would be lovely.
Okay, and one final question, which I think is fitting to close the Q&A session. How can we ensure that the benefits of microbiome research are equitably distributed, particularly in resource-limited settings?
So equitably distributed, that is a quite a difficult question. I think it's a very good question, but it's a difficult one for me to answer. I think I'll answer it with the following, is that we need to take it from the ground up. So that's where our SciComm people come in, where they need to help us be able to distribute our microbiome information in a palatable way to people who are not researchers. So you actually get, for example, in the faculty of molecular biology and human genetics, there is actually a scientific communication group. And this year, for example, we have formed a task team that will be working with a media company who make worksheets for science clubs in schools around the country. And the focus of this year is actually microbiome. So we'll be introducing the kids to microbiome through comic strips and easy activities through a worksheet. So that's kind of where it's a difficult thing, because we want to make sure it's available for everybody. But that's where we need to start roping in our scientific communicators, where they can help us get the message across. And I think we need to do this on as many free platforms as possible. Potentially, the web is one of the easier ways to do it. Going to schools these days to educate people is becoming a bit more difficult, because you actually need all sorts of clearances to do that. So I think using the web and maybe popular online newspapers like the Daily Maverick to write lay articles is a good starting point.
Okay, on that note, I would like to thank our presenter, Dr. Neil Van Zyl, for a very insightful session. I would also like to thank everyone who joined and participated today. We appreciate your participation and engagement. And to all participants, please just take note that the session has been recorded and will be made available on our website. I've just posted the link in both the Q&A and the chat. We really look forward to you joining us in our future masterclasses. Okay, thank you, everyone.
Thank you, Luther, and thanks everyone for joining.